So we provide bioinformatics sequence analysis web services. So I'll be describing this work today for you. So just briefly, an outline for the talk today. So I'll be describing job dispatcher framework. This is the framework that in which we provide access to core bioinformatics tools and data resources. So I'll give you a brief summary of the different bioinformatics applications that we provide and data sets, databases. And importantly for the talk today, I'll be describing the web services API and try to encourage you to use the API. And quickly go over and do a brief programmatic access demonstration and show you some example integrations and leave you there with some links. The talk kind of follows a similar outline as Youngmi's talk, so that's the approach. So here, this is the services page. When you go to EBI website, and if you go to services endpoint, you will see this sort of page. And you can see here there's a list of tools and data resources, and Job Dispatcher, this framework that I'm presenting to you today, provides all, not all, but most of the tools that you can use, and we provide web services for it. So I have to say though that some of the teams in some of the group teams in EBI provide their own access to tools. They provide also APIs, and for example, the case of Ensemble, they provide a REST for accessing the data that they provide. In any case, I'll be focusing on Job Dispatcher. So what is Job Dispatcher? As I say, this is a framework that we have been developing over the last 10 years or so. This is a Java application which allows you, integrated access to core bioinformatics tools. In a second, I will show you what sort of tools we provide. And we provide, of course, up-to-date biological databases. So, for example, if you want to do a sequence similarity search with, for example, BLAST, we provide updated databases for that. Importantly, we provide programmatic access, so you can sort of integrate these tools and data in your own analysis pipelines. And as hopefully I will be able to kind of demonstrate that it's relatively easy to integrate this into your analysis pipelines and workflows. And just to kind of give you a brief statistics, over the last year, 2017, we performed more than 140 million jobs with Job Dispatcher. So the sort of tools that we provide, these are bioinformatics applications. As you can see, we provide a lot of different multi-sequence alignment tools, as well as pairwise sequence alignment tools. For example, Clustral Omega, as well as Emboss, Needle, and Water. We provide sequence similarity search tools such as FastA, NCBI BLAST, SciSearch, and SciBLAST, for example. And we are in the way of integrating also AMR tools like PAMR. We provide also InterProScan and other protein functional analysis tools such as Inferno. And we provide also other Emboss applications for other sort of sequence operations, sequence analysis, and so on. So this is just to give you a view of how, for example, a web interface looks for a tool like Clustral Omega. Typically, you have a text box area where you can paste your sequence, and of course, in this case, because it's a multi-sequence alignment, you need to paste a number of sequences, or even you can actually do the same analysis using, for example, a session identifiers. And at the bottom of the page, which I'm not showing you, you would then just submit the job, and as soon as the job is finished, you retrieve the results. You can see, the different files that you have access. In this case, we would get, like multi-sequence alignment format file for Clustral. So in terms of data resources, we provide, of course, Uniprot databases as well as ENA databases, and we provide a bunch of other ones that most of them are coming from groups at the EBI. And so we provide, for example, sequences from structures from the Protein Data Bank in Europe, as well as, for example, Campbell targets and so on and so forth. And again, similarly, this is how user interface for a tool like NCBI BLAST would look like. In this case, you have here an input checkbox where you can select or unselect several of the databases that we provide. In this case, for the protein sequence search. And again, I'm not showing you here very well, but basically there you would type or paste a sequence for in this case to do a sequence similarity search using BLAST. So in terms of the talk today, I want to describe the work that we've been doing over the last months or so, which is to provide, similarly to what Youngmi was showing you before, we provide a Swagger user interface so that you can browse and navigate over the API that we provide. And for example, here in this case, we have a list of endpoints for a particular tool. So in this case, if you click on the drop-down menu there, you'd select the tool and then get to know and browse the different endpoints that you can use. So say that in this case, I selected Cluster Omega. The first endpoint here would be parameters. So if you try that, you'd get an XML list of the different parameters that Cluster Omega you can use from there. And also, for example, in the case that you get the list here, you can then get to know more details about each individual parameter. So we have another endpoint, which is parameter details, where in this case, you need to type one of the parameters that you got listed in the previous endpoint, and in this case, you would get to know more about this parameter. So in this case, for example, you'd get to know a description of what the parameter means, the typical values that you'd use, as well as, for example, the type of the data that you can use, if it's a string, if it's an integer, and so on and so forth. So we have the run endpoint, which is actually a post call. In this case, for Cluster Omega, there's a number of different parameters that you can use. You typically use, for example, default values, or you can use the ones that you can, for example, choose a custom set of different parameters. But typically, you would use an email address. You select a number of sequences for, in this case, running Cluster Omega, multiple sequence alignment. And for example, when you submit, you can actually test this here. You can submit a job, and you would get a job ID back from the call. And in this endpoint, then, once you have a job ID, you can do a status call to check whether the job is finished or not. So in this case, for example, you would get a return saying the job is finished or the job is still running. From that moment, once the job is finished, you can also use another endpoint here, which is result types, which would give you back a list of different results or the files that you've got for the job that you just submitted. So for example, in this case, again, since it's a Cluster Omega run, you'd get a multiple sequence alignment format file, and you would also get, for example, a dendrogram file, as well as, for example, a sequence identity matrix or such thing. So now I'm going to try and do a bit of a demonstration how you can use some sample clients. So we provide a number of sample clients to interface with the API. So this is here just the GitHub page for the group, web production. So the first repository here, which is called Web Service Clients. This is the repository in which we provide the sample clients. We provide sample clients in a variety of different languages. And I mean, we would be happy to actually get pull requests from people and submitting their own takes on how to put these clients together. And I'll just quickly put a video through. I'll try and explain as I go along. So in here, I'm just going into that particular repository. So here, basically, this link here points you to the Swagger user interface that I was just describing, so you can learn more about the endpoints. And as you can see, we have clients for REST and SOAP in a variety of different languages. And I would like to tell you that we also have now Perl clients in a Docker container, so you can simply download a container from Docker Hub like that, and straight away can start using the clients that we provide. In this case, we are using or focusing on the Perl clients here. And now I will show you briefly how we can actually start using the client straight away. In this case, using, for example, the NCBI BLAST Perl client. When I pass help, I will be seeing the command line interface help for that particular client. And in this example here, I'm actually starting to do a query, or to doing, in this case, a similarity search using BLAST. So in this case, I would be using the S-type, sequence type to be protein. A database, I would be using UniProt KB SwissProt and BLASTP, and this would be the query. This is SP SwissProt, and the query would be pack4 human. So I'll just quickly show you that. Those commands, in this case, because I've got the latest version of the container here, this step is fast. In your case, it might take a bit longer to download all the layers that it needs. But the good thing is that you don't need to install any of the Perl dependencies on your local machine or environment, so very easily you can. So the command that I was running there was this one, passing help as a parameter to NCBI BLAST, and you can see immediately the help for the options and parameters that you can have available in this case for this particular NCBI BLAST client. So the next step, what I will be doing is an example, as I said, an example usage for this particular client, using making actually a query or making a sequence similarity search analysis using BLAST. So literally just copy and paste the command there, just to tell that in this case we need to use a valid email address. In this case I already described that we were using SwissProt and BLAST-P program. And I'm also using here out format IDs just to get that particular file out of the computation. And as you can see, the job is started. We got a job ID now, and soon will be finished. That it is, and we got the file back that we specified the IDs. And what I'll be doing now is just literally getting the add of that file to another file, which I could have done more easily anyway. But that's what I was doing there, just kind of to demonstrate. Because now I can actually do a bit of an workflow. Now this add contains sequence of UniProt identifiers, which were the hits of the BLAST. And I can actually start, for example, running multiple sequence alignment on these hits. So that's what I'm going to do now. You can see I've actually, in this particular repository, we have a folder called util. We have some utilities there. And we actually have these Python utility to get sequences from these identifiers. So I'm just using the IDs.text and output. I will get its.faster file. In this case, I'm literally just getting sequences in faster format for this particular its. Takes 1 or 2 seconds. And in this case, I will just blast that file to confirm that, yeah, in fact, we got the sequences back. What I can do now is actually run cluster omega Perl client here with this its.faster, and immediately I just started the job. I'll get a job ID back, and as you can see, after a few seconds, we get the result, which in this case will be a cluster formatted file, which I will just show you right there. So in a similar fashion, so now I can actually go from this step and, for example, generate a phylogenetic tree. That's what I'm going to do next with another client, in this case using simple phylogeny Perl client. And you can see now, if I do just confirm that we in fact got, we got a cluster omega formatted file there. And now using the simple phylogeny client, in fact, in this case, I will be using just the actual job identifier from the previous step. We could use the alignment file anyway, but yeah, this has started, generates another job identifier and creates the dendrogram. And in this case, I'm just checking whether this is in fact a neoweek formatted file, and it seems to be. So it kind of, I really wanted to encourage you to use the web services. So I think it is relatively easy for you to integrate these tools, and the great advantage I'd say is that because we have such a vast amount of different databases, you can, for example, run BLAST against all these different databases. That would be, I mean, I think it's relatively easy for you to run the tools, but you know, you can run BLAST, but in order to maintain all these different databases, I think it would be great advantages for you to actually use our own services. So now, just giving you a few examples of integrations. So for example, UniProt, Protein Data Bank in Europe, and Ensemble Genomes, all of them provide in their interfaces a BLAST where users can run a BLAST. In this case, they are consuming our web services. And in this case, for Interpro, they also run Interpro Scan from our web services as well. They have that integration in their system. So lastly, just to give you some useful links, this is the repository I was showing you. We've got sample clients there, but this you don't need to use these clients. You can actually make up your own clients because the endpoints are quite easy to navigate, and you can see the full API user interface over in here. This is the main address that you would start to use the web services, and this is the link for documentation. You can read through the documentation, and of course, as Youngmi was saying, you can always reach us via the EBI support contact form, and yeah, we'd be happy to help you integrate the services within your analysis frameworks and pipelines. So finally, I'd like to thank you for your attention, and also web production team, Rodrigo Lopez, and our colleagues. So actually, just using the opportunity to say that we are currently performing some interviews. So if you are really keen to work in such projects, contact us, and we'd be really interested to hear from you. That's all from me. Thank you.
Thank you.